Hello everyone, I'm Jing Xing from Michigan State University. It's my great honor to share our recent study today. The title of my talk is Published Anti-SARS-CoV-2 in Vitro hits share common mechanisms of action that synergize with antivirals. I will talk about the following topics: the background of COVID-19 and anti-SARS-CoV-2 drug discovery, the common MOAs of some active compounds, the validation in patient datasets and in vitro, and their synergism with known antivirals. For more than 1 year before the vaccines are available to the public, COVID-19 continuously caused stress in our life. Every day, there are hundreds of new cases in Michigan and thousands in the whole US. It already caused more than 3.8 million deaths. So the research on therapeutics is of great importance for COVID-19 treatment and the future pandemic. Generally, intervention on any processes of viral life cycle might have some antiviral potential. There are some drug design strategies targeting on viral proteins, such as the RDRP inhibitor remdesivir and the viral main protease inhibitor GC376. Also, targeting the host cell is promising, such as the lysosome acidification inhibitor chloroquine and the cysteine protease inhibitor aloxastatin. So far, hundreds of active hits were reported, but most of them showed only moderate effect. So we further analyzed them to enhance our understanding of COVID-19. We mainly focus on the host cell transcriptome data interpreting because new variant viruses might be resistant to the current viral enzyme inhibitors. We utilized the LINKS dataset, which contains around 8,000 drug profiles of their effect on gene expression. And according to our literature survey, 97 of them are active anti-SARS-CoV-2 compounds at cellular level. We identified 63 differentially expressed genes favored by the active compounds and called them the anti-SARS-CoV-2 compounds signature genes. For instance, the NPC1 expression is generally induced by the active compounds with a very significant p-value derived from Wilcoxon rank sums test. And the MYBL2 expression is inhibited by the active compounds. We further validated the gene expression change induced by a positive drug, cephalosporin, in human lung airway cells. The NPC1 expression was induced 2 to 3 folds. Then we investigated the key pathways targeted by the active compounds using gene ontology enrichment analysis. 2 pathways are significantly enriched in the 63 DE genes. The cholesterol homeostasis with the 3 red genes mapped and the microtubule cytoskeleton organization with the other 8 genes mapped. Generally, 36 of the active compounds regulate 1 or 2 of these pathways, and these drugs' primary mechanisms are quite diverse, such as NF-kappa B inhibitor, selective estrogen receptor modulation, and histamine receptor antagonism, but none of them are antiviral drugs. Then we designed the ChoMCyto score to quantify the gene expression pattern involved in these two pathways, collateral homeostasis, microtubule cytoskeleton organization. Basically, we took the average of the upregulated genes and the negative average of the downregulated genes, then added them together. We found the pattern of ChoMCyto indicated COVID-19 patient severity. In the first patient cohort, we processed the transcriptome data of 33 PBMC samples. In the heat map, the left column summarizes the effects on ChoMCyto genes induced by active compounds in our collection. The NPC1, ECCB1, and HMGCS1 are upregulated, others are downregulated. But the comparisons between patients and healthy donors exhibited a reverse pattern on these genes. We used the ChoMCyto score to quantify this reversal pattern and found the ICU patient had a lower negative score than the severe patients. Also, severe patients scored lower than the moderate patients. There's another cohort with 65 leukocytes samples labeled with their sequential organ failure assessment or SOFA. We can see a very significant correlation between ChoMCyto and SOFA. Then we applied the ChoMCyto score to screen all 8,000 drug-induced gene expression profiles in links to evaluate their anti-SARS-CoV-2 potential. Monensin was one top candidate not reported before, so we tested its antiviral potential in viral E6 cells. It could inhibit viral replication with IC50 of 11 micromolar and didn't show much toxicity. Then we examined 3 positive drugs, cephalosporin, lomitapide, and monensin, on the human lung airway cells to see whether they regulated collateral and cytoskeleton pathways. All the 3 drugs induced the NPC1 and HMGCS1 expression, which are key genes involved in collateral homeostasis. For the beta-actin, a component of the cytoskeleton, it was not detectable after cephalosporin treatment, and its expression was also inhibited by lomitapide and monensin. Because viral infection involves many processes, drug combinations targeting on multiple processes might benefit. After calculating the ChoMCyto effect of drug combinations reported by Bobrovsky, we found the host targeting drugs with higher ChoMCyto score present synergistic effect with known antivirals. The Spearman correlation is 0.7, which is quite good. For example, the nitazoxanide, its profile clearly follows the pattern of upregulating these 3 genes and downregulate the other 8 genes, which presents a high ChoMCyto score. It also showed synergistic effect with several antidrugs like Abidor and remdesivir. Thus, we propose the future drug development should co-target viral proteins, collateral homeostasis, and microtubule cytoskeleton organization. To summarize, published SARS-CoV-2 inhibitors share common mechanisms of action. They regulate collateral homeostasis, and microtubule cytoskeleton organization. The transcriptomic pattern of these pathways indicates COVID-19 patient severity and anti-SARS-CoV-2 activity in vitro. Monensin inhibited SARS-CoV-2 replication in viral E6 cells with IC50 of 11 micromolar. Compounds co-targeting these processes might present a synergistic effect with known antivirals. This work is recently accepted by briefings in bioinformatics. With that, I would like to thank my mentor, Dr. Bin Chen, for the support and suggestions on this work. Thank all the collaborators from MSU, Institute Pasteur Korea, and Spectrum Health Hospital for the bench validation and medical advices. Thank you all for your time. I'm glad to take questions.